name is Rita Vassena. I'm a scientist by background, and I am currently the CEO of a small startup called Fecundis. And this is a Spanish-based company that is trying to improve the results of in vitro fertilization treatments. The main problem that we are trying to address is the very high inefficiency of this treatment. We know that if we start from 10 oocytes, for instance, we usually get no more than 1-2 high-quality embryos, and of those, approximately only 1 in 3 will give rise to a healthy pregnancy and a live birth. What we are trying to do is to improve these low odds of success. And specifically, we are working on the semen side because with our technology, we have found that by manipulating precisely certain biochemical pathway in the sperm before fertilization, we can have embryos of higher quality in higher number, and they tend to implant better as well. If we look at this from a clinic point of view, there would be a first step of sperm selection. And then we add in our own technology that we call it HyperSperm, which is a series of steps by which the sperm transits through a series of media that have proprietary composition, of course. And this mimics little by little the same steps that are happening in the female reproductive tract. And so that at the end, the sperm is ready to be used for fertilization. So these are incubation, subsequent incubation in different media with different compounds. And then it's used like for fertilization as in a regular cycle. There is no other difference in terms of operation for the clinic. So it's not a selection tool. In fact, sperm needs to be selected before we apply our technology. But apart from that, we do not modify the sperm. We simply put it in the best capacitating condition possible. So as you know, capacitation is a biochemical process that the sperm needs to undergo in order to fulfill its full potential in terms of biological capacity of fertilize and drive very early embryo development. And we have observed that in our clinics, capacitation is not really paid much attention to and is not really done properly. So what we are trying to do is to change this and improve capacitation to the maximum extent possible. So we have collected very extensive preclinical data in mice and also in cows. In mice, we went to the 2nd generation. So we have data of safety on the pups that are born after HyperSperm, their birth weight, the growth curve, their fertility to the 2nd generation, and everything seems to be normal. The embryos that are developing are normal, and they have, in fact, of high quality. They do cluster in the high range of morphological quality. Eventually, we will have to make sure that this is conserved in human as well. Nevertheless, all the tests that we have done so far in terms of safety on the sample after use of HyperSperm, for instance, DNA fragmentation or motility or vitality or survivor curves, they all report a very, very, very high safety profile with no really difference from the control. There are already several babies that are born out of the clinical trials. There are 3 in the initial pilot of just 10 patients, and we have about, I think, 16 pregnancies that are now ongoing in a second larger clinical trial. So none of these pregnancies seem to have any particular issue, not in their process of the pregnancy or in the health or weight or anything else of the babies that are born. And I think this is most reassuring and is something that we are really very happy to see. We are actually testing this. The technology is undergoing clinical testing. As far as we can tell, there shouldn't be any specific limitation in terms of, for instance, concentration or motility of the sperm sample within a certain range. What we do know, what we do not know, because we have not tested it, but it's that this technology, we don't know if it would work in sperm that is testicular or epididymal. So far, everything that we have done is with ejaculated sperm, and it has worked quite substantially the same, irrespectively of the quality of the sperm sample. So I think for a long time, when dealing with the sperm sample, we as a field have focused on having, at the end of the selection process, let's say, a pretty sperm sample, right? That moves correctly and is mostly motile, without debris, without other cells. We try to focus on the actual function of the sperm. In both preclinical and a pilot clinical trial, we have found that if you process sperm with HyperSperm at the end, you have more embryos at the blastocyst stage. And these embryos have a very high morphological score. So they cluster into Gardner score that is AA usually. If you translate this to the clinical process, what we can predict it as most likely, it would lead to shorter time to pregnancy and higher pregnancy at the first transfer, which I think is a very worthwhile clinical outcome, I would say, to pursue. Well, there are some technical challenges, of course. Every time that something is translated from a very controlled environment to a real life clinical environment with the variability that we have in our species, there's always some noise that come up, and it's very easy to go from super happy when one patient responds well to doubting everything when one patient does not respond well. So I find that in order to be in this type of environment in a startup trying to develop a new technology, you really need to kind of hold steady emotionally for every day, basically. Another challenge, which I think you will hear very, very often, is the challenge of funding, right? Until when you are in the market, you're just spending money, you're not making money. And so you're very heavily reliant on funds, either diluting or not diluting. So that's more or less my full-time job, finding funding so that this project can move forward. So right now, what we have so far is a pilot in a split design pilot with 10 patients, so 10 controls and 10 treatment, because of it is like a sibling oocyte. This was done under very controlled condition with donated oocyte and normal zoospermic sample of sperm. Right now, we have a second larger clinical trial that aims to recruit 40 couples with a much wider inclusion criteria. So women up to 40 can be included. The oocyte are from the patient, not from a donor. And also the sperm quality can vary down to 3 million per ml. So it can be really relatively low in terms of concentration. The goal of this second clinical trial is to try to understand whether the technology as it is developed now does apply to a wider spectrum of patients in a little less controlled and more noisy background, so to speak. But also one of the goals is to see if there are some subgroup of patients that may benefit from some slight, adjustment in the technology. And that, to me, speaks to the personalization and the, personalized caring of patients in today's IVF field. Well, we are still finalizing our path to market. We are still validating some of the hypotheses. I don't think that we will be able to be in the market for sure before 2026 and perhaps a little bit later, but that's our goal. We are trying to keep an open mind because we have so many people that are engaging in R&D. So we think that we may have an interesting pipeline. However, as you probably know, we are a small company, a few people, and we really need to take on one large project at a time. So for now, we are focusing on this one. Yeah, it's a very exciting moment, I think, to be working on the male side or sperm side in reproduction, especially human reproduction. That is, we feel so much to do and so little already done that I think we will be busy for a long time.